This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 504, recorded on July 27th, 2018. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today here in New York City, Dickson Despommier.
Hello, Vincent.
So what's it doing out the window? Cloudy, right?
It looks like it's going to thunderstorm. That's what it looks like. Hot.
Yeah, I just got a severe thunderstorm warning.
Yep.
It's currently 31 Celsius.
It's going to change.
It's going to go up or down?
Down, way down.
Also joining us from Western Massachusetts, Alan Dove.
Good to be here.
Hey, Alan.
It is tropical.
Yes.
In Massachusetts.
For a week, yes.
Soon they'll be having yellow fever.
Well, I have been seeing a lot more bugs because it's been pouring rain every day and humid and sticky, and it's 89 Fahrenheit, 32 C right now. And we are expecting afternoon thunderstorms.
Right.
Also joining us from Bend, Oregon, Rich Condit.
Hi, everybody. How you doing?
Rich is on vacation, and he's joining us. Thank you.
Do retired people take vacations? I don't know. I'm still learning about this.
We take works, not vacations.
It is 77 degrees here. That's 25 C.
And not humid, I bet.
The humidity is 20%.
Right.
Wow.
Which means the dew point is 34 degrees. We are headed for 92 degrees today, but the nighttime temperature overnight, it will go down to 55.
Nice sleeping, as Ronald Reagan used to say.
It's awesome.
It is.
And Rich just texted us a photo of where he's sitting in his little cabin. He's looking out over a river.
Not just a river. It's a world-famous river. It's the Deschutes. The Deschutes.
The Deschutes. Did a river run through it?
It is a river.
Yes, and it does run through it, but I think that's a different location.
There's a big Blackfoot River in Montana. That's a different river.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, everyone. So the rain has made it all the way to New Jersey.
I told
it's actually raining pretty hard right now, and I can hear a lot of thunder.
Do you have a window? Can you see out?
I can see out, yes. It's raining really hard.
Outstanding.
It's 87 or 30 Celsius with lots of rain. So I guess you'll be getting it soon.
We will.
Yeah, we've had a lot of rain in the last few weeks, right?
We have.
And it's unfortunate. Well, in the middle of ASV, about halfway through it was really warm, and halfway through it rained, and it kind of cooled things off for a while.
Yeah.
I was thinking today that it felt like the same heat and humidity that we had at ASV.
The Viruses and Cells Gordon Research Conference needs your financial support. NIH and foundation support has declined dramatically in recent years. The organizers of the 2019 meeting, Julie Pfeiffer and Britt Glounsinger, need $70,000 to support partial registration and travel costs for 32 speakers, 18 discussion leaders, and selected students and postdocs. You can donate. I will provide a link in the show notes to the donation site. Your donations are tax deductible, and you'll be acknowledged as a friend of TWiV, unless you don't want to be, of course. You could be an enemy of TWiV.
How about a neutral party of TWiV? We
just returned from 8 consecutive road TWiVs. This is amazing.
Yeah.
That's 2 months.
2 months.
Back in 496, that was in May, Rich and I. Vaccines Work from Irvine. Then we had 497 was Europic from the Netherlands. 498 from Atlanta, ASM Microbe. 499 in Halifax, Canadian Society for Virology. 500 in Austin, Texas. 501 in Texas A&M. No, 501 was Outbreak in Washington, D.C. at the Smithsonian. 502 is Texas A&M, and 503 last week, ASV. Some of these, the order of some of these is not related to the actual order.
Yes, there wasn't actually travel from Austin to Washington and back to Austin for Texas A&M.
No. It's kind of like a paper, the way you read the paper is not how the work went.
Correct.
But now we're back in studio for about 2 months, I think, the usual. And so here we are. And we have some follow-up from last episode, which was at ASV, from Steen. Dear hosts, I thoroughly enjoyed meeting most of you in vivo at ASV, having listened for nearly 4 years. Svetlana's and Anne's enthusiasm on TWiV 503 was great. I realized that the first all plant virus TWiV thing was good-natured ribbing and an ongoing joke, but since the topic arose, I wanted to push back by listing just a few of the great plant-centric episodes, arcs you have recorded. TWiV 402 and TWiV 70 on host manipulation by cucumber mosaic virus to attract vectors and pollinators. TWiV 343, which featured counts of infection foci on leaves analogous to the lesions on Chenopodium species, such as quinoa, that Anne described. This episode also featured jokes about turnips.
A joke about a turnip?
How could
Tell me a joke about a turnip.
Well, 2 turnips walked out of a bar.
Really?
It can happen.
Right.
No, I just thought they would turn up their nose at anything they would put in front of them.
That's going to be hard to beat.
Good.
Ooh.
TWiV 459, 403, and 47 on producing vaccines in the silencing immunocompromised host. Nicotiana benthamiana also mentions in the episode. I look forward to more episodes featuring plants, insects, and fungi on this and other microbe TV shows. Literal-mindedly yours, Steen. So Anne ribbed us about that being the first. It may have been the first with all plant virus guests, right?
Yeah.
Yeah.
But we will have Anne back regularly, once a
Great.
month, hopefully, to talk about plants.
This is a pretty comprehensive redux of plant virus TWiVs.
Yeah, he
Yes.
did a great job. Steen is
I
outstanding.
couldn't have done this.
Steen is wonderful. He's a wonderful listener, and he took pictures of us after TWiV.
Yeah, it was good to meet him. Hi, Steen. Nice to put
Sorry,
faces to names.
He's not far from us in Cook College,
Tell him to come on over.
which is in,
Bring him on.
what's in the town in New Jersey?
Rutgers, New Brunswick.
New Brunswick. Is that New Brunswick?
Yeah, it is.
If you drive down Route
I just did that.
1, on the right you'll see the sign, and then there's cows behind it.
That's true.
It's an ag school?
Yeah.
Mm.
It's one of the first in the country.
Yeah, well, his advisor, Siobhan Duffy, we should have on.
Right.
Because maybe Dickson, you and I can make a field trip. Well, then we can't have the Skype, so.
No.
Apparently her mom lives up here somewhere near Washington Heights, so
Oh,
get her to come over.
that'd be good.
Alan, could you take Juan's?
Sure. Juan writes, Dear Vincent, Rich, and Kathy, hosts of episode 503, The Greening of TWiV. As a plant virologist and big fan of TWiV, I want to thank you for this wonderful all-plant virus episode. However, as a faithful listener from the very early episodes, I can tell you that the appearances of plant viruses at the show, on the show occurred more often than you may think. I started to download and listen during my commute to work every week since 2011, and I recall vividly several episodes where plant viruses were definitely among the stars of the show. Here's a non-exhaustive list. Episodes 24, 79, 92, 155, 268, 271, 272, 334, 343, 347, 402, and 469.
Hike.
Yes. Anyway, if you cannot increase the number of episodes, which will be great indeed, we'll keep listening and learning things from you. So thanks again. And Juan is at the Center for Research in Agricultural Genomics in Barcelona, Spain.
Cool.
Barcelona.
I note that his list of episodes is almost completely non-overlapping
Yes,
with Steen's.
Mm-hmm.
So there's been a lot of plant virus episodes.
there have been.
We'll have more.
And that certainly occurred to me when that line came up in that episode, too. I was like, wait a second. We've talked about plants.
Hang on.
Anne was fooling around.
Yes, of course.
It's good that she has a sense of humor.
Indeed.
You know, since it was a live show, you really should have had them sitting in the audience and said, we're going to take volunteers to be guests on the show, and then they would come up, and so they'd be plants.
That would be funny. Oh, that would be very good. Plants, get it, Dickson?
Yeah, you know who else has a good sense of humor is orthopedic surgeons.
They do?
Sense of humor.
Humor.
Oh.
Yes.
Arg.
They like breaking your bones.
They do.
That's
All right. Now, about once a month, we're going to have a special segment.
Oh.
And this is related to Outbreak, the exhibit, which the American Society for Microbiology is proud to support. It's called Outbreaks, Epidemics in a Connected World. This is at the Smithsonian National Museum of Natural History, now through May 2021. 3 years. Here at TWiV, we're excited to see the topic of zoonotic viral diseases getting such a public platform. As a tie-in, we'll be discussing information on one of the viruses featured in the exhibit in a special segment each month. This month is on Nipah virus. If you're in D.C., be sure to go see the exhibit. It's free. And, of course, don't miss TWiV 501 Outbreak, where we went inside the exhibit. We had to start filming at 7:30 in the morning.
Oh, before
people came in, and that means Ray Ortega and I went at 6 a.m.
Lord.
to start setting up. But there were people there at 6 a.m. They're working people. Did you get to see it?
6 a.m. is a wonderful time of day.
It was fine.
I haven't seen that episode yet.
A cup of coffee, and it's fine. Brianne, did you go see it?
Did. I saw it about 2 weeks ago, and I really enjoyed it. I also recently learned that they have the DIY segment, where you can actually get a lot of the materials to set up an exhibit where you are. And I just got an application yesterday to try to put together a version it drew.
Cool, yeah, so we mentioned on that TWiV, the curator, Sabrina Schultz, said most of the exhibit is available as a digital download. And so you can download it, print it out as big as you want. You can modify it to make it. So she said if you are in Bangladesh, you could modify it to include more of Nipah if you wanted to, and so forth. So, yeah, it's really cool. And I'm going to do that for my course next semester as well. All right, Nipah virus first identified in Malaysia in 1998. It was an outbreak of respiratory and neurological disease in pigs and humans. The outbreak started in the Perak state, clusters of cases of encephalitis among pig farmers. And they had contact with the pigs who were sick. They had respiratory illness and encephalitis and deaths. And this spread south to the major pig farming areas and peaked in April 1999. And to stop this epidemic, they had to cull over 1 million pigs. And if you go onto YouTube and search for Nipah pigs, you will find videos of them digging big holes and just pushing the pigs into them. Really sad. I mean, they couldn't, I guess, individually inject each pig with, something, but they buried them alive.
These were live pigs they were putting in?
Yeah, because you can see them walking around and the bulldozers are pushing them in the hole.
Oh, wow.
And then they cover them with dirt. It sucks.
That's not good.
I showed it to my class one year and they're crying.
Wow.
No. But, if they're sick and you go to inject them with a drug, you may get infected, I guess.
So I have another approach.
Or if they could have done whatever procedure you normally use to slaughter them, that's going to have risk of infection as well, I guess.
No, it would have been easy to do. You cover them with CO2.
Yes.
Maybe they did that. And I
cylinders of CO2.
Yeah, I didn't
fill up the pit after they get in there.
I didn't see, but I know they push them in the pits with bulldozers.
Horrible.
Or you don't even have to have cylinders of CO2. You could just hook up a car exhaust and get them with carbon monoxide.
Either way. Oh, yeah.
1 million pigs is a lot of meat.
That's a lot of meat.
A lot of pigs.
Yeah. So the virus was isolated in March 1999 from the cerebrospinal fluid of a patient with encephalitis, and Nipah is named after the village.
Right.
Nipah, capital N, for those of you pedants.
A lot of viruses are
which
named
is not
after
in
villages. No, it's not.
Nor is it in Connecticut.
Norwalk, man.
And it was subsequently isolated from the urine of Malaysian flying foxes.
Which raises a question here.
Yes.
When bats eliminate, do they have two separate openings for feces and urine, or is it a single opening?
I believe they have two because they're mammals.
Right.
Why do you ask?
Well, because I have to change my...
Bat's diaper?
No, I have to go back to the beginning and change my joke then.
So John Epstein, who is part of the EcoHealth Alliance here in New York City, also part of that is Peter Daszak, whom you know,
right.
Dickson.
I do.
John was a science advisor for the Outbreak exhibit. He was on that TWiV 501. And if you look in some of the links we're going to provide later, you'll see pictures of him holding flying foxes in Bangladesh. So he went there to work. We're going to have him on TWiV sometime. You could talk about his experiences and all of that.
Right. He's an interesting guy.
So they stopped that initial outbreak by culling the pigs, but there was another outbreak in Bangladesh in 2001. And since then, there's been an outbreak almost every year and a couple in India. And in Bangladesh, the vector was date palm sap.
Right.
They, and at the Smithsonian exhibit, they have a palm tree with, someone collecting date palm sap. They put these open containers on the tree, and at night the bats would come because they like sap as well.
Syrup.
And they would eliminate, as you would say, Dickson, and contaminate the sap, and then people would drink it and get sick.
Bats do indeed, I just searched this up, bats do indeed have the same excretory structures.
Okay, not like birds.
And
not like birds.
The solution,
or lizards.
So at the Smithsonian, they actually have a few of these date palm sap containers, and the Robert Costello, who walked me through the exhibit the day before, said they were very expensive to acquire. They came from Bangladesh. Yeah, and so that by putting a cover on them, that stopped the transmission because the bats couldn't get into it. Sure. And they have some of the covers there. I think they have a photo of some of the covers in the exhibit. And they have a huge model of a flying fox hanging from the ceiling.
They're huge.
They're big animals.
Huge.
Yeah.
And they also, down further in the exhibit, they have a couple of actual specimens. They're not alive, of course, but they're part of the museum's collection.
Right.
Which they drew upon for this exhibit. They pulled out the bats and other things, mosquitoes, reduvids, and
when you're the Smithsonian, you've got a big closet.
They have 140 million objects.
Yep.
Wow.
In their collection. That's incredible. Over at the American Museum.
How many of them are beetles, though?
Many. And they have an inordinate fondness.
And the rest are nematodes.
A picture of me is in the collection.
Really?
Yeah.
Cool.
Picture of me pipetting at MIT. Because it was part of an exhibit at the Museum of Natural History.
Oh, that's fantastic.
On polio, and it's there as a young man with no gray.
You've been immortalized.
Awesome.
Now, I have a link to a very nice review article on all aspects of Nipah and Nipah virus infection by Ong Lim and Linfa Wang, the original Batman.
Mm-hmm.
Nipah virus infection, the Journal of Clinical Microbiology, which you can have a look at. I believe it's open access. It might not be. It's hard for me to know because here I click on it and I get right in. But there has been a recent outbreak in India, which we talked about on TWiV 495.
Kerala.
What's that?
It's a region of.
Yeah, say it again. I like that.
Kerala.
Yeah.
It's open access.
Yeah, it is open access.
Thank you very much. And in conjunction with that, I received an email from Gopal, who writes for, he's a science journalist who often writes for various publications in India. He's written to us before, and he wrote, I think it would be worthwhile devoting a TWiV to the current Nipah virus outbreak in Kerala state, India. It looks like the outbreak is subsiding. I hope I'm not speaking too soon.
Right.
And he sent a bunch of links.
Yeah.
And one of them is an article from Tufts. Tufts Now, why do we need to worry about Nipah? And the picture right there on the top is John Epstein in Bangladesh holding a rather small flying fox.
Baby.
And he's got gloves on and a mask and glasses and a cap, but there's no TWiV on the cap. I got to get him one. And that's a nice article which goes over the outbreak. Now, the interesting thing, as Gopal says, Nipah outbreaks have occurred in Bangladesh and the neighboring Indian state of West Bengal, but this is the first time such an outbreak has happened in Kerala, a state in the southwestern corner of India, far away from West Bengal and Bangladesh.
That's right.
That, it seems to me, raises the question of how it suddenly manifested here. So, yeah, Bangladesh is up in the upper right-hand area, and Kerala is way down at the left. It's far.
Maybe they imported their palm sap.
I don't know. But we're going to.
So where again were the former Indian outbreaks? They in West Bengal,
right across the border from Bangladesh, right?
Yeah.
Okay, so this really is geographically a big difference.
It's huge.
About as far apart as you could get.
Correct.
It's about from New Jersey to Texas.
Maybe even further.
I don't know. He goes on, strangely, just 2 outbreaks were reported from West Bengal between 2001 and 2012, while neighboring Bangladesh had several episodes.
Yeah.
One suspects that Nipah cases occurring on the Indian side of the border may be getting missed. Now, I have no idea what it's like in West Bengal. Have you been there, Dickson?
No.
It may be quite rural, in which case you would miss cases, right?
So named, by the way, because of the tigers.
Yeah.
So it's quite rural.
Wait, so how were the tigers named?
They were named after the district. No, I don't know.
We're going around in circles here.
You remember the story of the tigers that turned into butter, don't you?
No.
Oh, yes.
No, I don't.
Oh.
That's an old Southern story that's definitely not politically correct.
Well, it's an Indian parable first.
It is, yeah. It's actually originally derived from probably a Choctaw or Cherokee legend, but
so Gopal sent a number of links, WHO articles summarizing the outbreak. There is one interesting article in the Hindu News, which is the anatomy of an outbreak. It's quite long and interesting and talks about how it occurred, how it was detected, how they responded to it. Quite interesting, which maybe most people would normally not see. But in this level of coverage, you would not see, I think, in a publication outside of India, most likely. So it's really nice to read. And then The Economist had an article about a nurse who died on May 21st of the disease, taking care of a patient who was ill. And this is a typical way it's transmitted, is to take care of someone with either within your family or in hospital and not have proper protection, and you get infected. But I found an article in SIDRAP which says that the transmission patterns have been analyzed. So there were 19 infected patients, and 17 of those appear to have gotten infected from an index patient, a 26-year-old man. And the people exposed included 3 family members, 4 people at the first hospital that treated him, and 10 at a medical college where he was taken for a CT scan. So he infected 17 people.
Yeah.
So I was wondering if that was
killed. He didn't just infect them.
I know there's been human-to-human transmission in the past, but this seems like a lot.
It says the 17 out of the 19 died.
Yes.
17 out of 19.
That's an incredible mortality rate.
That's higher.
It's been between 75 and 90% in the past.
That's higher than rabies. No, not higher than rabies.
So here's the key here. Health officials suspect that this first patient got infected from fruit bats, and that he and his brothers might have been infected after being in a bat-infested well, although there were no fruit bats in the well. So I'm not quite sure.
Well,
what that
right.
means.
So this one of the stories that Gopal sent is a very, very thorough. Which one is it? I think it's the anatomy of an outbreak.
Yes.
Story from
Hindu News. Yeah.
Hindu News, and that goes into quite a bit of detail about the story and the patient 0, who fellow named Sabith. Or Sabbath, and he was a 26-year-old plumber and was cleaning out a bat-infested well, apparently, with somebody else in his family. So then, after all this, and this virus basically wiped out the family,
right?
And a
so
bunch of people at the hospital as well.
Is the idea that he got infected in this well?
And the idea is that he got infected in the bat-infested well, but when they went back to investigate, they only found one bat, and it's an insectivorous bat that's not a fruit bat. So they think that wasn't it. But I'm wondering, if they were cleaning out the well, maybe they got all the fruit bats out of there, and that's why you didn't find them. And then some random bat happened into it and was unrelated to the story, and that's what they found there.
So I guess we can assume that there are fruit bats in this area, right?
The review that you posted from Journal of Clinical Microbiology has a really nice map with the distribution of the bats that spread Nipah.
Right.
And so it does show that area.
So there are fruit bats, and maybe there's also Nipah in the bats, although I don't think anyone's looked yet. They may be looking now. So then the question is, why now? And, presumably there's been contact with bats over the years. But then you could also say in 1998, why then, right?
Exactly. Right. No, you've got lots of these open-ended questions.
One of the other things I found, I think it might have been one of these WHO documents or something linked therein, talked about the overall ecology of this, and the key thing that you need for Nipah transmission from the reservoir is human contact or or pig contact or possibly other intermediate host contact with the bats.
Mm-hmm.
So why do you get bats in with the pigs or in the well? It's because they don't have their normal habitat available to them in the woods.
Right.
So they're going to go where the fruit is. And if you've cut down a lot of the woods and developed it, then they're going to be moving into where the humans are keeping their fruit and their fruit trees.
That's right.
So a lot of this is development driven. There's also the surveillance problem that in the past we hadn't really, probably a lot of people died of Nipah virus 50 years ago, and we just never knew what Nipah virus was and happened to discover it in 99. But the seasonality of it probably has a lot to do with development patterns and human development patterns and also drought.
That's right. And Kerala is one of those states that gets periodic floods and droughts. I mean, they're really inundated. It's one of the big failure agricultural areas in India, and is responsible for a lot of hyper-urbanization of people actually moving out of those areas. But I think the popularity of mangoes has something to do with this also. Because mango, the pig farmers realized in Malaysia that if you cut out an area of, let's say, forest, or call it jungle, and planted mango trees in the periphery, the mangoes could supply the food for the pigs. So they had a double crop going, right? Except that it also attracted fruit bats.
Yeah.
And the original outbreaks, they actually observed this happening. A fruit bat was eating a mango that had fallen down onto the ground, and it was identified by a pig, and the pig rushes towards the bat to see the fruit, and the bat panics and, of course, it urinates on the
and the fruit.
And then flies off, and now the pig grabs the fruit and develops the virus.
Yeah.
As the pig gets sick, the farmers started to panic because they thought their pigs would die, so they drove them to market early. And that's how they started this epidemic.
Yeah, pig farmers also got sick.
Yeah, and they got very sick.
With the pigs,
And the pigs will die too from this if they don't sell them. So if a sick pig is noted, it has about 3 or 4 days after that before it dies, as I recall, and they then take them to market right now.
Yeah, there's something, one of the local terms for this is barking pig syndrome. The respiratory aspect of the disease gives them this whooping cough.
Sure.
Cough.
That's right. It's related to limited agriculture, consumer demand for a new fruit, in this case, all kinds of different mangoes.
But in this place, Kerala, right? Kerala. There are no pig farms. This is bats.
That's right. However, I've been to India many times, and there are tons of free roaming pigs.
Yeah.
There is a S-load of free
feral pigs.
Okay. Not so feral. They're peridomestic. They're not wild, but you see them in all the canals.
But they don't raise them for food, obviously.
They don't raise. Well, there's a subset of people in India that do eat them.
I see.
There are,
but not on the level of Malaysia, where millions.
No, that's right. You know what they're doing? They're for the tourist industry.
So they walk around.
Because they'll slaughter pigs for the people that come in from other places, right?
Like you, right?
Like me,
right?
Right.
By the way, the distance from Bangladesh to Kerala is in fact about the same as the distance from New Jersey to Texas.
That's right. That's right.
Nice.
It's about we're talking
1,500 miles. That's right. Exactly right.
I mean, if you look at a map, there's India. It's tiny, right? But no, it's not.
Right. But we're talking about a reservoir host that has a range that's that big.
They call India a subcontinent. It's that big.
So the bats could travel from West Bengal or Bangladesh.
I don't know.
Certainly their ranges.
But they need to eat. They're 1,500 miles worth of fruitless.
Yeah, and I don't think so. I don't think we're looking at an individual bat traveling from New Jersey to Texas. But if you have overlapping populations and it's a flying species, they're going to have a contiguous coverage of this virus, and they're a reservoir host. They don't seem to get especially sick from it.
Right.
So they're just carrying this thing around with them wherever they are, and their distribution extends to both of these places.
Alan, I don't think they're reservoir hosts. I think they're the natural host, and therefore they are resistant, just like the African birds are resistant to West Nile virus. Those are the natural hosts.
What is the prevalence of this virus in the bats? Do we know that?
Peter Daszak might know the answer.
Yeah, well, we'll get John on the show and ask him. I'm sure it's.
I think it's known, but
yeah.
But as Alan was saying, here's a paper: prevalence of Nipah in
flying. Oops, I hear thunder.
I'm seeing the lightning out the window.
Yeah.
Oh, it stopped here.
Fort Lee is getting creamed right now.
Okay, can I back up with a quick correction?
Yeah,
you bet.
Sure.
The story of Little Black Sambo, which was the one Dickson and I were talking about, I accidentally mixed that up with the Uncle Remus stories, which are completely different origin. And the Sambo story is actually Indian, not Cherokee and Choctaw.
I meant to say something.
May or may not even be derived from an actual legend.
No,
but
maybe Kipling had a role in it. Jungle Stories or something like that.
Might be both Scottish author.
Don't you hate it when they don't put the final numbers in the abstract?
Then you have to read the damn article.
Well, for here doing this, I don't want to take the time to read it, but there have been studies done. Right. We can look at that. But the other thing I thought is that this person might have traveled to West Bengal.
Totally
somehow
possible.
Contacted about it and got infected and then went back, but apparently he didn't. One of the articles I read said he traveled 8 months previously, but that's too long before to have done anything. Right. And so that's probably not it. So we're probably talking about local virus that's just manifesting.
And I think the story about the bats in the wells in the well is probably significant.
I suppose.
I agree.
Because it's the right time frame. It's bats, and the fact that they didn't find fruit bats in the well that they'd just been cleaning out just suggests maybe they did a good job cleaning it.
I think a bat infested well is a good title.
Yes,
a bat infested well.
I have a title related to that.
There is, by the way, there's this article which I referred to before in Tufts Now. It's basically a U.S. perspective. Do we need to worry about Nipah virus? And they have a photo of a date palm sap collector using one of these bamboo skirts to cover both the top of the collector of the sap and the bark on the tree, because they shave the bark off the tree, and of course the bats would go to that too and contaminate it. Then it would drip down even if you had a covered pot. So they cover both. And they also ask, is there a risk of Nipah traveling outside of India? And John Epstein says, someone could get infected, then get on a plane and go somewhere else.
Right.
Wasn't Nipah the sort of idea behind the movie Contagion?
It was, yes. And they say that in this article, actually. Yep. However, that was a little bit of a different virus.
Well, sure.
But this is as
Contagion.
Right.
They made a hybrid of two different viruses.
But as this points out, and this infection highlights, it can spread outside of, this is not something where you need a particular mosquito to spread it, or you need blood contact. Yeah. Here we're talking about a case where one person got sick and went to the hospital, and the virus spread to their whole family and wiped out a bunch of people at the hospital.
Right.
Yeah. They note in this article that India is really on top of this, and they make sure that they do case tracking and they limit travel so that you limit the spread.
But there was also a healthy amount of skepticism in a lot of the coverage from the Indian publications in particular about how the Indian government has had a hit or miss history in keeping track of infection rates.
A couple of other interesting answers here. Is there any positive news? Yes, the government knows how to deal with it. The Indian Institute of Virology in Pune has done research, and Pune has done research and diagnostics on the virus, and India has good public health infrastructure. An investigation was launched quickly. The country has the resources to contain it. Then this is an interesting one. How do public health researchers track the disease? The research primarily implicates giant fruit bats, which travel quite a long distance to feed at night, up to 50 kilometers from their roost. This means you don't need to find giant fruit bats roosting where people were initially infected. They could have flown in from somewhere else to drink fruit, to eat fruit or drink sap. That's why adequate wildlife testing requires an understanding of bat ecology and behavior. It's not simply a matter of grabbing the closest bat to the house of the infected person. I like the word grabbing. Is there going to be a bat backlash in India? We hope not. We're encouraged to see many Indian news stories reminding readers that bats are both not to blame and are ecologically important. And we stress that spillovers are rare. Yeah, don't go out and kill bats.
No.
So I found some information on prevalence here from I pasted this link in at the bottom of our discussion, right above the email, if you want to look at it. There's an article in Emerging Infectious Diseases from 2001, and there's a table in that article where they've surveyed 237 wild-caught Malaysian bats and looked for neutralizing antibody status to Nipah virus. And basically, depending on the species, they found anywhere from 0% up to as high as 31% in one bat species. So the 4%, 5%, 31%, 17%, a bunch of zeros. They also looked at some insectivorous bats, and they found one bat infected out of what looks to be maybe 40 or so sampled, so a lower percentage in that species. All right, so it's not 100%. It's but it's not zero. It's we're talking maybe 5 to 10%, depending on the species,
and
maybe even as much as 30.
And the species they found the highest percentage in, Pteropus hypomelanus, is the small flying fox.
Now there is there's currently no vaccine, although on TWiV 495 we talked about an organization that is spending $25 million to help develop a vaccine, and this is supposedly consisting of the viral glycoprotein. By the way, we haven't mentioned that Nipah virus is an enveloped negative stranded RNA virus.
One of 7.
And it's paramyxovirus.
Paramyxovirus.
Same family as measles, mumps, rabies, but it's a subfamily that's it's got the same basically genetic structure. Subfamily that also includes Hendra. And Nipah and Hendra are pretty closely related and have a similar sort of story, I think, where it's a natural infection of a bat that spills over into either directly into humans or into another species, and the humans acquire it, and there's some human-to-human transmission as well. And of course, in the alternate host, it wreaks havoc as opposed to the non-clinical infection in the natural host.
So they call them Nipah viruses.
Hendra
Nipah. And Hendra is not one of the viruses in the exhibit, unfortunately.
Well, you
By the
can't
way,
fit them all.
if you want to know what the viruses are that we'll be talking about besides Nipah, MERS coronavirus, SARS coronavirus, hantaviruses, Ebola viruses, HIV, influenza, Zika virus, and smallpox.
Roger that.
Roger that. That's what we'll be doing every month or so. We have 3 years.
Sounds exciting.
So I have a question. Let's say, first of all, they don't even know what the correlates of protection are for Nipah virus. So we need to know that, right?
Right.
Absolutely.
Because you can't say, let's make a vaccine that induces antibodies, because you don't know if that's...
Has anybody been infected twice?
Somehow, as I recall, there was really good evidence that this G glycoprotein vaccine is effective, and maybe in animal models or something like that.
Yeah, there are good animal models.
And the cause of death is an encephalitis?
Yeah.
Yes.
Yeah.
Has anyone been infected twice?
I think the numbers are pretty low, so it's hard to...
Yeah.
I don't know the answer, but, in this outbreak, 17 of the 19 died.
That's incredible.
So you can bet those 2 others are going to stay out of trouble. But it's a good question.
There going to be donors for antibodies.
So who would get this vaccine, assuming it's developed at some point?
Question. I think anybody who farms pigs.
Yeah, well, the pig farmers should get it. Malaysia, right?
And perhaps the pigs.
Yeah.
Well, yeah. In the case of Hendra, as I recall,
you vaccinate the horses.
One of the strategies is to vaccinate the intermediate host, the horses.
Yep. Right.
A true One Health strategy.
That's it.
Which is a big focus of the outbreak exhibit, by the way.
In the case of Hendra, that's essentially the same concept in the vaccine, right? The glycoprotein?
Yes.
Yes.
So, yes, pig farmers would get it, but obviously you wouldn't give it to everyone.
Health workers.
In India. Health workers could get it. And then, of course, if there's an outbreak, you immediately do a ring vaccination.
Ring vaccination.
Right? Exactly right. But herd immunity doesn't exist, by the way, Vincent.
Yeah, right. Yeah, we got an email about that.
That's right.
Oh boy.
By the way, one of the other aspects that the Indian press cover particularly well is the doctors on this case, and how alert they were to the possibility of Nipah virus and unusual viruses emerging, and really sharp medical care probably made this a much smaller outbreak than it could have been.
Yeah, they did a good job.
Yeah, one of the interesting things about that is that the doctors recognized that this was not your standard encephalitis. In particular, since there were multiple family members presenting with the same symptoms of encephalitis. It looked like an unusual type of encephalitis that was transmitted from person to person, and that set off alarms for them.
Yeah.
So the cellular target for this virus is multiple, because this might be respiratory as well, right?
Yeah, it is, for sure. Yeah.
Right.
So
some of that was in that review as well.
Yeah.
It was talking about how broad the range of hosts and cells were that this virus could infect.
Because you make a short list of encephalitis-causing viruses, and you're stuck with EE.
Well, it's not so short, actually. Measles. The measles is one of them.
Yeah.
Which is a paramyxovirus. Mumps.
You always get encephalitis with measles?
No, of course not.
Well, that's what I'm saying. The ones that you always get encephalitis from.
Well, I don't think
so. West Nile, Eastern, and
West Nile doesn't always cause encephalitis. 5%, 5 to 10% of the
but it goes to. Even though you don't get sick, it still goes there. Though.
Well, that's not encephalitis if it's not causing inflammation of the brain, right?
All right.
If it's just replicating without inflammation, without overt symptoms. So no virus is going to do 100%.
All right.
But
equine encephalitis does always, right?
I don't think I wouldn't say any virus does it all the time. No.
So they're not 100% neurotropic.
No, why would you be? Why would you go in the brain 100% of the time? You'd never get anywhere as a virus, right?
You're trying to think like a virus. They don't have a brain.
No, I'm not thinking that.
They have your brain.
You know I wouldn't do that.
I know, but you just did.
This is anathema. But in terms of evolutionary selection, as I've said, the main pressure of selection is for a virus to find a new host.
Yeah, but remember,
if you go in a brain,
we're an aberrant host.
Yeah, for what?
For all of those viruses that you're talking about.
Yeah, you're right.
So
we are aberrant, period.
So you forget about whether it's an evolutionary significance or not.
You're leading me down all different trails.
No. But the natural
good to have
cycle.
You back, Dickson.
No, you're welcome. The natural cycle for West Nile is birds.
Yes, we're an aberrant, but now it's a human virus.
And they don't die.
It spreads from human to human via mosquitoes.
This is true.
Anyway, what was I saying before?
I have no idea.
The last thing I wanted...
You needed to have some stumps ground.
Did you need some stumps ground?
Welcome back, Alan.
So I found one last thing. I looked for some recent research that might be interesting, and I found a paper on trying to find out if this virus is transmitted by aerosol. We know it's transmitted by close contacts with pigs or infected people,
right,
consuming date palm sap, right?
Right.
Is it an aerosol transmission? So in this paper, very interesting, Journal of Infectious Diseases, just published recently, they have a model where they infect Syrian hamsters. And they've used them before, but this time they put them in a nebulizer chamber, and they delivered virus to them. And of course, this is done in a BSL-4 laboratory.
They're nebulizing the virus, not the hamsters.
They didn't nebulize the hamsters, and they say aerosolized virus caused similar clinical manifestation of disease, lesions in the lungs and the brains, which are the two main sites of virus replication, when infected by aerosol as compared with other, inoculation methods. So it could be transmitted by aerosol. And I would bet that, if a bat sneezed close to you, it might be able to transmit it as well by aerosol.
Especially if you're a hamster.
Yes, right. Anyway, that's Nipah.
That is Nipah.
It's supposed to be 15 minutes, but we can't
we
do
can't
anything
do that.
else.
This was 15 minutes.
We can't do anything for 15 minutes.
This is our 15 minutes.
That's right.
Well, I enjoyed, refreshing my memory on this. Of course, my first, the minute a virus comes up, I have to remind myself my first stop is viral zone.
Yes, of course.
Because I gotta put it in a family, and I gotta see the genome structure, and I gotta think about all of that. And then my second question in this case was, Nipah, Hendra, I get them mixed up. All right. Because the stories are so similar and the viruses are so similar. So I had to kind of confirm that and put it together. And all these resources and that one article that discusses, that Alan's been talking about it, that discusses in detail the investigation. I hope you'll link that in the show notes. That's a terrific article.
So Australia doesn't have any Nipah.
They have Hendra.
But they've got loads of fruit bats. They've got a huge load of fruit bats.
Mm-hmm. I noticed that when I looked at the map of where these bats were distributed, that there didn't seem to be any reason why there wasn't Nipah in Australia.
Right. Or Papua New Guinea.
So they have Hendra. Yeah.
Papua New Guinea. You think one excludes the other?
Or for that matter,
Great question.
Why is there no Hendra in India?
Wouldn't that be interesting, though?
Right, exactly.
What if you could substitute Hendra for Nipah in Malaysia, for instance?
Doesn't seem like a trade up.
Well, what does Hendra do to people?
Encephalitis.
Same thing.
Same stuff.
Except you get it from
stories
horses.
are very similar.
Yeah, horse trainers were getting it from the horses who got it from the bats.
Were they dying?
Yeah.
They were dying, okay.
In fact, years ago, we talked about this. It was an outbreak of Hendra, which happens regularly still in Australia, although now with the vaccine there shouldn't. And one of the veterinarians who was involved in the first case actually emailed us and said he'd be happy to come on the show and talk about it. And I never followed up.
So it would be interesting to know whether, for example, the replication in the intermediate hosts is actually somewhat different for these two. In other words, if Nipah has a preference, say, for pigs over horses, then that could explain what humans experience as a difference in the geographical distribution. It could be that it's kind of where the pigs are, as opposed to where the horses are. But I don't know. But I think both viruses will infect both animals.
Yeah,
both have a very broad host.
I mean, in this case, it's not so easy for the viruses to move from continent to continent, right? If it were mosquito-borne or human-transmitted, effectively, no. But here, unless we bring a flying fox to Australia from Malaysia, it's going to be hard. It may happen at one point, right? But
and for anybody listening, don't do that.
Don't take bats.
But it can only be transmitted if bats are incontinent.
Yes.
Great.
So I need to
dying to say that.
Rich, no. I just thought of that. Rich, I must ask you a question. Why did you refer to a host as intermediate host?
Well, okay, so
because I think that's in parasitology talk.
You've got a real
stage.
Definition for that, right?
Well, for parasites, yes, but do they refer?
Right, I even think of the reservoir and natural host as the same thing.
But it's not. It's not.
I know.
In parasitology, what's an intermediate?
An intermediate host is a host which harbors a developmental stage of the parasite in anticipation of the final definitive host, which harbors the sexual stages of the parasite. Okay, so it's a totally different concept here.
Right. I'm just I'm not using formal definition of the word.
So what is the natural host for Hendra and for Nipah?
Yeah,
the natural.
As far as we know, bats.
Okay, so we're accidental hosts then.
Yeah, we are.
Okay, fine.
We are spillover hosts.
Let's go spillover accidental.
Eventually, the infection, the chain of infection stops,
right?
Mostly because we intervene,
except for these 17 poor people.
But an interesting question would be, if left unchecked in any way, would it continue? And I would say no, probably not, because these things have been happening for years. Most likely, we just never saw it. And this is a virus that just peters out, just like Ebola. Eventually, will peter out.
I was just going to say,
and you need a new spillover to start another chain.
That's right. That's right. I mean, even when you're talking about a mosquito vector for a virus, that's not an intermediate host because the virus still replicates it the same way as it does in a normal, in the natural.
Are there some cases where the mosquito is the definitive host?
Correct. That's Plasmodium.
It's the definitive host for Plasmodium.
It is.
But for a virus, say.
Is there a virus of just mosquitoes? Of course there is.
Must be.
Have to be.
And
they're sopholoviruses, so they're probably are mosquito viruses.
Yeah.
Have to be.
And they're transmitted vertically, I would guess.
Maybe.
Right?
Yeah. I would think so.
So you would like it where there's no mammalian host required or intermediate required for the transmission of that mosquito virus, right?
Yeah. I mean, it's
Yeah, I'm sure there are, and we've probably talked about it, and we'll get email reminders.
Don't you remember episode? Yeah,
they could catch it by drinking plant juices, and that could be the source for their viruses.
Could be.
I'm sitting here wondering if mosquitoes sneeze,
and fell to his knees, and what became of the monk?
Let's move on.
Indeed.
We have a couple of interesting email, and we won't get through them all.
Boy, don't we!
But the next one will probably use up most of our time. This is from Clarissa. Dear Vincent and TWiVers, last Monday, now this is many Mondays ago, because we haven't had a
many Mondays ago.
The WHO confirmed circulation of vaccine-derived poliovirus type 1 in Papua New Guinea, one case of acute flaccid paralysis and detection of virus in stool samples from other children. After 18 years without a case, vaccine coverage is only 61%. That's too bad. Brazil used to have excellent coverage rate above 90 to 95%, but now it's around 77% is for polio vaccination. Actually, Brazil used to have an excellent vaccination program. All vaccines for kids are part of the national vaccine program and are free of charge in Brazil.
Wow.
But coverage rates are dropping every year. Let me just pause here. And this is brilliant. All vaccines are free of charge. Right. If Brazil can do this, why can't we?
Brazil actually invented.
No. I can tell you why we can't, but that's going to be a whole different.
It's not a word can't.
I love when Alan starts.
It's just why don't we? Not why can't we? Because we could, but we don't. So why don't we?
Yes.
Why don't?
If I'm not mistaken, Brazil actually invented the concept of a national vaccination day.
That's
where they go through a huge campaign to advertise this and then vaccinate everybody in the country on one day.
That's amazing.
Well, I do know that Albert Sabin went there in the late 70s, early 80s, and did massive national immunization days with polio vaccine and showed that this could work to bring down the incidence dramatically, because previously people thought, well, you can't do that. You have to immunize everyone at birth. You can't just go in and give it all. And he said, no, you can. And he showed it in Brazil. So maybe they had been doing that. And
Brazil, by the way, has the best mail system of any country. They know where every single person.
Do they have a good female system?
I'm not going to answer that question. They can find out where everybody lives because they all have a mail address. And so they actually have programs of public health. They actually tested out whether you could use drops of blood on filter papers for diagnostic tests by mailing them around to people and then making sure that it lasted long enough to validate the test, and they could.
It wouldn't work in this country. People would claim it's too invasive.
Yeah.
All right. So Brazil is having an issue also with, this is a problem which we'll get to, but as soon as there's no polio was eradicated from the Americas, right? Many years ago and other WHO regions. And then the countries say, it's gone. We don't need to.
They get complacent.
Yep. All right. Continuing with Clarissa, I wonder if you guys could discuss the challenge to eradicate polio using IPV and OPV.
Oh, boy, can we.
And the decline in vaccine coverage over the years in several countries. We have, in fact. In Brazil, in addition to the economical problems that affect public health, science, and education, the change from OPV to IPV, bivalent OPV, clearly parallels the lack of adhesion by the population. I also think that pediatricians and primary healthcare workers have not been so demanding on the importance of vaccinating children in general, maybe because they don't see too much of these preventable diseases. A weird way to forget about the importance of vaccination. My understanding is that the future scenario to eradicate polio is complicated. That's perfect.
It's an understatement.
I understand that the use of OPV will never eliminate poliovirus from the world because virus will always be excreted in stool of vaccinated children. However, if we use only IPV, the excreted vaccine-derived polioviruses from previously OPV vaccinated children and even wild poliovirus imported from other countries such as Afghanistan and Pakistan will circulate silently, particularly if vaccine coverage is not too high. This is a fascinating discussion, and I would like very much to hear the opinion of the TWiV people.
Quite a pickle, isn't it?
It's a pickle. Clarissa is at the University of Rio de Janeiro in the Institute of Carlos Chagas.
Great institute. It's a marvelous institute.
Chagas of Chagas disease?
You got it. Absolutely.
I think basically Clarissa summarizes the problem pretty well just in her letter.
Yes.
It does.
And let me embellish, and Alan and I can embellish, and anyone else who wants to embellish. So when this happened, this Papua New Guinea case, I blogged about it. It was a couple of weeks ago. It was June 27th. Now, previously, just a few weeks before, there was supposedly a case of polio in Venezuela, which was also alarming because, again, the Americas had been declared free. There hadn't been a case there for 30 years. And it turned out, now I've forgotten the reasons, but it wasn't. It wasn't. But this one is clearly vaccine-derived poliovirus. I think those kids in Venezuela had acute flaccid paralysis, and there just happened to be poliovirus circulating.
Right.
But the sequence indicated that it was not sufficiently diverged to have caused their paralysis. But in this case, vaccine coverage went down. These vaccine-derived polioviruses circulate for sure many years after you do immunization campaigns. And if coverage goes down, you get outbreaks. And that's what happened in Syria last year. We had like 90-some cases of type 2 vaccine-derived polio because the immunization rates dropped to like 60% in Syria. So this is a problem because we're vaccinating now against the vaccine. The wild polio cases are 30 last year, mainly in Pakistan and Afghanistan. So what do you do? Well, WHO has said, let's switch to IPV, bivalent OPV. But that's a problem because you have to inject IPV. And that's why, as Clarissa said, vaccine coverage goes down. That's part of the reason people just don't like needles. It takes a trained healthcare person to deliver it. It's problematic. And then if you have a case like this one in Papua, what do? They go in and do mass immunizations with OPV.
Right.
And you start the problem all over again. So at some point, you have to bite the bullet and just do IPV. I don't think with a needle that's going to work. I would love if we had a microneedle patch IPV ready to go.
Yeah.
And that would be so much easier to deliver, right? You put it on with a Band-Aid and you don't have to have a needle and all the problems associated with that. I think. And
that could pave the way to many, many more vaccines getting to many areas that can't currently deliver them.
Yeah, it'd be like a DNA chip. You just have 15 vaccines in the same.
These are in testing at the moment, and the testing will not be as extensive as normal because we can't do an efficacy test. We're going to just be able to say, do you get a similar immune profile as with the other vaccine? And then we have to license it because there's no other way to do that. But I think, in fact, at Europic, I asked my panel of 4 picornavirologists. First, can we eradicate polio? 2 of them said yes, 2 of them said no. And I said, if we did, should we stop vaccinating? And all 4 said no.
No.
Mm-hmm.
But if you have no cases of polio one day, how can you keep vaccinating the entire world? Their countries will not go for that. As you can see already, they're not going for it. Right. So that's the problem. So
and the sales job for this has had to evolve over the years because the initial hype for eradicating polio by the year 2000, which was what was going on when I was in graduate school. That was the story.
That's right.
Was we're going to wipe it out by the turn of the century. So the part of the way they were packaging that, the CDC and WHO were packaging that and selling it to leaders of countries around the world was, look, after you eradicate this virus, then you don't have to keep paying for vaccination. And some picornavirologists at the time pointed out that this is not a good way to sell that because it's going to run into a whole bunch of problems like the ones we're seeing right now. And here we are.
So, I mean, I think we're going to have to continue with the current regimen. Now, we've removed type 2 OPV from the preparation because type 2 was eradicated. And if we keep using type 2, we keep making more cases of vaccine-derived type 2. And that's last year in Syria, there were, as I said, 90 cases. So we've removed type 2. The vaccine now consists of type 1 and type 3. We haven't seen a case of type 3 in ages. And so I asked Mark Palanch of the CDC, are we going to make a monovalent type 1? He said, no, it's too complicated. If you keep giving countries new vaccines every so many years, they don't like that. And they actually stockpile the old ones because they think they must be valuable if you're telling them not to use them anymore.
Right.
So weird things happen.
Exactly right.
So we're going to be using this bivalent. And I would say that if we have no more wild polio, then everybody's got to start using IPV at some point. But the coverage is not going to be as high, so we're going to have outbreaks.
And you hit a separate problem if you do get to a world where you've eradicated the virus from the wild, and you're giving everybody IPV, and let's say the OPV from chronic secretors is finally washed out of the system. We're really, really rid of polio worldwide, except that we have to grow huge, huge batches of live virulent virus in order to manufacture IPV.
Yep.
And then we're going to have another discussion about how to do that, because if you want to stop vaccinating, you're going to have to either move that into a P4 manufacturing facility, which I can't even imagine how much that would cost, or you're going to have to you're going to have to develop an entirely new vaccine that's going to be maybe subunit based, and go to something like that. And this whole idea of stopping vaccination, that was what we did with smallpox. And now, of course, we're right back around to spending billions of dollars developing new smallpox vaccines and stockpiling existing ones because we're concerned that somebody might release the stuff or it might be accidentally released. So I don't think you ever really get past that kind of a problem.
At Europic, I learned there are actually quite a few vaccines in development. Besides the microneedle patch, there are actually new OPV strains, which supposedly do not revert. And these are being actually tested in humans. There's a facility in the UK where they put these containers. What do they call them? The transportation containers that you put on the back of a truck. What's the name for those things? Those big metal things. If you drive past the airport on New Jersey Turnpike, you see they're all stacked up. Shipping containers.
Oh, shipping containers.
So they put them in the parking lot, and they made this, what do they call it? The polio villa or the polio spas. And they recruit volunteers, and they give them these vaccines. They have already been immunized already, and they challenge them with vaccine strains. So there's some development, there's some hope that they can be used, and then that will get around to having to grow them in a P4 facility. Right. There are also empty capsid vaccines that are being developed, which would not need a BSL-4 facility.
Yeah, and we've discussed those, vaccines that are modified viruses that are avirulent that you could still immunogenic that you could use for IPV.
Yeah. I should point out that the WHO does have an end game strategy. So I'll put a link for this in the show notes. It's a comprehensive plan, it tells you where they're going to go. It involves interrupting all polio transmission first, and then withdrawing oral polio vaccine. And so I think that's the plan at the moment. And it goes on until you get these cases here and there, and it gets interrupted because then you have to go back to using extensive OPV to contain these outbreaks, right? But WHO is very clear. This is our strategy. We're going to pursue it, and they're not going to get distracted by these outbreaks. And hopefully they won't become more numerous as we get closer and closer to real eradication.
Yeah, they do have a plan, but you can reach your own conclusions after reading about whether it's a sensible plan.
Yeah. Sorry, Brianne, what were you going to say?
I was going to ask whether we know if the polio vaccine gives any cross-protection against other enteroviruses. Because isn't there an idea that with stopping smallpox vaccination, that might be related to some of the monkeypox outbreaks because it was leading to some cross-protection?
Right.
Rich, I'm waiting for you to...
certainly in the case of smallpox, I think that's...
Yeah.
I don't know how good the data are, but it is true that the smallpox vaccine protects against monkeypox, and withdrawing the smallpox vaccine would create basically a human a naive population for monkeypox. And I think that certainly that's been talked about as part of the reason for increasing monkeypox. Whether the same holds true for the poliovirus vaccine, I have no idea.
I'm not aware of any cross-reactivity in that sense with other enteroviruses, but there are lots. There are many, many out there. There's some major ones that dominate, like enterovirus 70 and 71 and 68, and there don't seem to be any protection by polio antibodies. But it's actually something this came up at Europic, and someone, I think it was Eckhard Wimmer, said, if we stop vaccinating at some point in the future, we will have a world in which no one has antibodies to poliovirus. And that has never happened probably in human history.
Right.
Because even before the vaccine, people got infected very early in life. And he said, just think of that. And if that's a good thing or not. Right. Who knows?
Well, we're doing the experiment with smallpox. You know, I mean,
right.
We're approaching a world. I wonder, you know, what the prevalence of antibodies to smallpox is in the population. Probably close to zero. Whereas it was, I'm sure, very high before eradication efforts or before immunization efforts became available. It's hard to imagine. I mean, and the planet basically doesn't have any smallpox anymore, except in freezers here and there, formally
or in the odd laboratory drawer.
Right. But boy, it's hard to imagine a planet without polio, isn't it?
Yeah.
Yep. Well, we'll see.
And it seems to me, you know, the WHO, the plan makes perfect sense. The problem, as always, with all of these things is getting people to cooperate.
Right.
Right. We were talking about how there's a Hendra vaccine for horses earlier. And I think I've recently seen something in the news about people not wanting to vaccinate their horses against Hendra, even though it's a pretty awful disease in horses.
Because the horse will develop autism, don't you know?
We have an email. We have an email about that. Actually, we have a listener pick. Yeah, people in the UK are worried that their pets are going to get autism from vaccines.
So sad.
What did Mark Twain say, Dickson?
What?
About common sense?
Oh, it's the most uncommon thing in the world.
Most uncommon thing in the world.
This is common sense. That's right. So another caveat here, of course, is civil unrest and just open warfare. That's where these infections pop up the most frequently.
Yeah, in Syria, that's been the cause.
Let's try to eliminate that first and see what happens.
I don't know. What's easier to eliminate, hatred or viruses?
Neither
viruses.
The answer is
viruses.
none.
Yeah.
I think viruses. Yeah.
Well, yeah, sure. I mean, that's true. But yeah.
All right, let's move on. Take a few more emails here. Rich, can you take Bob's email, please?
Bob writes, what is the difference between HPV and HIV? Science, in quotes, has been hijacked by political ideology movements. Scientists must appease the academic complex to get grants. The academic complex is hijacked by political ideology movements. This is all counter to the objective scientist model. In my humble opinion, your TWiV is a conduit of Trump hate and bitterness. I hope
that was in quotes, by the way.
Yes. I love your podcast, but it makes me sick when you deviate to political snubbing, hate, vitriol, and gossip. You become stupid, untrusted puppets. Why not tell us the difference between HPV and HIV?
I wish this guy had an opinion.
Did you not hear Trump and Gates discuss this? There are no dumb questions. I have always had comfortable conversations with scientists in the R&D workplace, in school, and my family. They don't prejudge me. They present knowledge in an interesting way. Your team's behavior is now the norm. That is sad. You're between a rock and a hard place. I still have writings, videos, audits of Richard Feynman to hold on to. So this email goes a couple of different places. I don't know whether the question about the difference between HPV and HIV is serious, but I want to answer it anyway.
So can I just jump in? I know where that question comes from.
Okay.
There was an interview where Bill Gates talked to Donald Trump, and then Bill Gates was talking about it afterwards, and he released the fact that one of the things that Trump asked him was, what is the difference between HPV and HIV?
Twice asked him in the same conversation, and was confused. He was unclear on that there was a difference. Like, are these even different things?
Yes. And that did take on a little bit of a viral character on
what did Gates say?
Internet.
We don't know that, actually.
We don't know.
I wish we knew, because that's the wrong person to ask.
So we brought this up. It was my fault. It was a pick of the week, and I brought it up over 2 months ago, which is the last time we had a pick. And so I did make fun of this, but the more important part of the conversation was that Trump asked Gates whether he should convene a panel on vaccine safety. I thought that was the more important part of that conversation, and I had meant to focus on that. So, yeah, I don't mean to mock him for wanting to know the difference, but the key was that he wanted to know if he should investigate vaccines, which is absurd. And Gates, to his credit, said, no, don't do that. They are good. Okay. And that was really what I wanted to focus on. And so I don't want to be a conduit of any kind of hate when politicians of any kind question science or make incorrect statements. I want to correct it. And I agree that we should do it in a way that does not convey any kind of hate. And I'm sorry that was the apparent.
It's too bad that Trump chose
effect.
Gates then to ask that question to, because Gates is not a scientist.
He knows a lot of things.
He does, but
I heard him talk at ASM.
It doesn't take you don't have to be working as a professional scientist to understand the public health ramifications of very different viruses. I mean, these are two things, and these are things that Gates's foundation has been heavily involved in. Vaccine distribution and HIV prevention, and so he is somebody who's on top of this.
He's a big funder of the polio eradication effort.
It doesn't mean that he knows everything about what his foundation is doing. That's the point.
But I've heard him talk. I heard him talk at ASM Microbe 2 years ago, and he mostly talked about global infectious disease. He was remarkably astute. I was impressed. He sat up front with an interviewer.
I'm not saying he's stupid at all.
He doesn't know the details like we do, right? Because he doesn't do it.
Right.
But he could tell him the difference between HPV and HIV.
So I have a question to ask them.
And so this guy's right. There are no dumb questions. I don't mean to make fun of that. I didn't like the vaccine questioning, which comes from not looking at any of the information about vaccines.
Yes. Doesn't this raise a problem, though? The problem is that Trump has never appointed a science advisor.
Yeah,
the science advisor could have answered this question in 3 seconds.
Agreed.
Yes,
it's true.
That's the reason why I'm raising it.
Well,
there is a long-standing antagonism here between Trump and his political party and scientific consensus on a huge range of issues. And my understanding of Vincent's pick was that this is touching on that point.
Right.
I would point out to Bob that he's engaging in exactly the type of ad hominem attack that he's accusing us of.
Exactly.
He might want to back off on that in the future. So just keep that in mind if indeed Bob is actually listening to the show.
We're actually not making fun of we're not making fun of Trump through hate and bitterness. We're not even making fun of Trump. We're accusing him of omission of sins that every other president has gone to great trouble to identify a person of stature to use as an advisor when these technical questions arise. Mm-hmm. And if you're not willing to listen to the technical answers, then why do you even ask?
Rich, what were you going to say about HPV and HIV?
Should we answer that question?
Go ahead.
HPV is human papillomavirus. Which, in terms of the virus itself, is a small naked capsid, double-stranded circular DNA genome virus. It's a virus that infects the skin mostly and comes in about over 200 different varieties, causes warts, is one of the common manifestations, but the reason we care about it is that there are several subtypes that can infect the epidermis, the skin in the cervix, and under some unusual conditions ultimately lead to cancer. And it is the cause of cervical cancer. There's a vaccine against it that can prevent cervical cancer. So that's HPV. HIV is human immunodeficiency virus, which is a completely different type of virus. It's a retrovirus, so it's got an RNA genome that's copied into DNA and integrated into the genome. In the virus, it's got a capsid and a membrane. It looks completely different. The life cycle is completely different. And HIV infects immune cells and ultimately, over a long period of time, kills them, and that's the cause of AIDS. Tough to vaccinate for. Actually, that's a big difference between the two, as we've developed an HPV vaccine that's extraordinarily infectious and extraordinarily safe because it is just the capsid for the virus. It has no infectious material in it. HIV is a sneaky sucker and is changing all the time, even in an infected individual, and it makes it extremely difficult to create a vaccine.
So I vote for Rich as Trump's science advisor.
Yeah, works for me.
Good with that.
Dixon, can you take the next one from Anne?
Sure. Anne writes, Professor Racaniello, so the rest of us are left out of this. I heard you say in response to a letter on the Immune podcast that you'd talk about politics when politicians negatively impact science. I completely agree with that sentiment. So here's another reason to talk about politics. And then she gives an article in Mother Jones about the National Science Foundation investigating. No, I haven't read this article.
Four GOP senators want
to
investigate the National Science Foundation because they found out to their horror that the NSF encourages science education.
Yeah, there you go. That's right. They want
to investigate.
And you know what? When they do, they're going to find out that's exactly what they do.
Yeah.
Mm-hmm.
Yeah. Oh, by the way, the National Science Foundation encourages science education.
Yes.
I should hope so.
Yes.
Well, they're
so the senators, just for those of you out there who are listening and have the misfortune of having one of these one of these individuals as your representative in the Senate, they are Ted Cruz from Texas, James Inhofe from Oklahoma, James Lankford also from Oklahoma, and Rand Paul from Kentucky. And they wrote a letter to the Inspector General's office protesting the fact that they found out that the NSF has granted $4 million to programs that encourage TV newscasters to discuss climate science. So they're
that may include radio shows.
That could, yes. So it doesn't include us. I know
I've tried, but I'm going to try again.
But they are distraught that the NSF is funding public education on science.
I get to vote in the election where Ted Cruz is up for re-election to the Senate.
Mm-hmm.
Yeah, as someone said at ASM Microbe, what do we do about this? Elio said vote.
That's right.
Mm-hmm.
Now,
exactly.
I wanted to say that this is NSF, of course, is a nonpartisan agency, right, and they're being investigated for supposed partisan bias, which, of course,
you know what? They're encouraging the education of people with regards to evolution, and that's definitely
they're encouraging. What they're doing here is they're supporting more discussion of climate change, which presumably these senators want to believe is not happening despite the evidence.
Well, I had the privilege of attending a meeting in Washington, D.C. before I went to TWiV 500, and it was sponsored by the United States Department of Agriculture, and the head of that agency is from Georgia, and his name is Sonny Perdue, that's his name. And every official from the United States Department of Agriculture that got up to speak started their presentation by saying the thing that faces agriculture most in terms of a negative is rapid climate change. And they shouted it out almost. So that's a government agency that's totally aware of the fact that this is an important issue.
The Defense Department is keenly aware of it as well.
Yeah, right.
Yeah.
It is a major security issue.
And NASA, the head of NASA was converted by the data from NASA. They said, okay, you don't believe it? Let's sit down and I'll show you the evidence and you can tell it for yourself. And he came out and said, you know what? They're right.
So just for kicks here.
Yes, as the science advisor, Richard, please speak up.
No, I'm just looking at real clear politics. I'm just going to present some facts here. Real clear politics on the Senate race for Ted Cruz and the Democratic opponent, Beto O'Rourke. And the Real Clear Politics gives averages of various news polls. So their average for the dates covering from May 19th through the 7th of July has Cruz ahead by 8.4 percentage points.
That's not much.
So this is... As these things go in Texas, Beto O'Rourke is giving Ted Cruz a run for his money.
Right.
So those of you who have an opportunity to vote, pay attention to this.
Yeah.
Makes a difference.
The other senator mentioned here, Rand Paul, I found recently an article, which I showed in my keynote, where he said he does not believe that vaccination programs should be mandatory, something to that effect.
Right.
Right. Let me quote it exactly. Let me find the slide. And
Isn't he a physician?
Yeah.
Yes,
he is.
Used to be.
Used to be.
Non-practicing physician.
I showed this because it was one of the slides that I said, this is one of the reasons why you need to. Here we go. Ron Paul is against compulsory vaccinations.
Ron or Rand?
Ron.
Rand. Oh, Ron.
Ron is against compulsory. Okay. Different Paul.
Different person.
The dad.
Different Paul.
But
the
still,
dad, that's right.
But still, the only way public health measures work is if everyone, Dickson, if you don't like toilets, can you go use outhouses, right, and get hookworm?
No, well, that's still a toilet. Can I go under a tree is what you should ask.
Yeah, right. All right, so let's continue. Just a few more. Brianne, can you take Ian's?
Sure. Ian writes, in episode 495, mention was made that a plant seems to have no motive to attract hyperparasitoids, as if this would reduce the population of parasitoids. See, Ian found a plant episode.
Right.
This could reprise the earlier comment about Ebola virulence. If the parasitoid is prone to unstable populations, the plant being able to control its population by hyperparasitoids could modulate the virulence of the parasitoid in order to promote its even distribution. Perhaps this is not likely, but it is a fun thought. Enjoying the show, Ian, from a very sunny Scotland at 18C.
So the hyperparasitoids kill the parasitoids.
Mm-hmm.
Right? If I remember that correctly.
And the parasitoids kill the
plants.
Well, they kill the caterpillars. Right?
They kill the caterpillars that are feeding on the plants.
Right.
So the plants should be pro-parasitoid and anti-hyperparasitoid.
That's right.
Right. And the plants produce a compound that attracts the hyperparasitoids, which seems counter to their interest.
Right.
But Ian's argument is perhaps this keeps the parasitoids from having a boom and bust population cycle.
Yeah.
So the plants want to maintain some level of the caterpillar population to keep the parasitoids alive, and to do that, they're attracting the hyperparasitoids. I mean, to the extent, as usual caveats on personification here, that plants don't actually want anything.
That's
but
right.
The evolutionary balance would be favored by doing this. It is a fun thought.
No,
yes.
Alan, plants do want something. They want to be a lawn.
Oh, yes.
Alan, can you take the next one?
Yes, moving on. Jean-Francois writes, hi, quick note regarding episode 494. You might have corrected this in 495 and received tons of emails about this already. RVSV Zebov, the Ebola vaccine, was created by scientists at the National Microbiology Laboratory in Winnipeg, Manitoba, Canada, which is part of the Public Health Agency of Canada. It was then licensed to New Link Genetics, which licensed it to Merck in 2014. Yeah. Regards, and Jean-Francois is from McGill University.
Right. So we said, it was a Merck vaccine, but they didn't actually develop it, but they made it and distributed it. But the development was absolutely in Winnipeg. It's right. We have to give them credit.
Thank you.
Tom writes, Vincent, regarding a frequent TWiX discussion topic, here's further discussion, some history regarding research publication. Tom in Austin. Tom, how are you? We saw you in person. And this is an article in The Guardian, why thousands of AI researchers are boycotting the New Nature journal.
It's a really good article.
Yes, it's very good. $3,000. You can publish in this journal. And so this is a good article, lots of background on this, and your publication in journal, which we talk about, publications in luxury journals, which we talk about recently. But what they did here was that a number of these researchers in this field got together. How many of them? 3,000 researchers signed a statement refusing to submit, review, or edit for this new journal, which is wonderful, but the other 3,000 in the field will go ahead and use it, so it's not really useful to boycott, right? Because someone's always going to publish.
The thing is, what I get from this journal is that these AI scientists to date have all published entirely open access, basically run their own show, and nobody pays for anything, and everybody communicates. And now Nature moves into the Nature conglomerate moves into the thing and creates their brand name title and starts a problem.
Yeah.
The cost of so many of these open access journals to publish in is really difficult.
Extremely.
Yeah.
I mean, open access is great, but $2,000 to $3,000 is absurd. So this is very interesting. I just finished reading Harold Varmus's book, the science and politics. No, the art and politics of science. Thank you. And he has this chapter on publishing, and because he talks about why he started PLOS, right? And he says it's completely fine for these journals to charge these. It's built into the NIH budget. It's a cost of doing research. Of course, he would say that because he started PLOS, which is built on page charts.
He's also the head of NIH.
Right. But I just don't agree with it. I don't think you should pay to publish your science. There has to be another way. I realize that that's the model, but...
That's fine if you're well funded or if you're getting funding from the NIH, but people who are at smaller institutions or who don't have a lot of funding can't afford that.
Everyone should have the opportunity to publish, right? It should be equal opportunity. But I
benefit a lot from open access publications since we don't have subscriptions to everything in our library here, but I can't afford the cost to publish open access myself.
Yeah.
Right. And the fact that NIH pays for this does not mean they should.
Also true.
Yeah.
It would be interesting to know more about exactly how the AI researchers support their publications. I mean, the problem is
they're currently building a bot to figure that out.
Right.
Yeah, the problem is that publishing stuff does cost money. Now, if you have a small field, you can do it all by, essentially some sort of fancy blog. Maybe that works. I don't know, but
it does cost money, but it doesn't cost as much as some of the top journals are. I mean, you have to bear in mind, and I'm a little constrained here because we're talking about one of my occasional employers is Nature, and I don't want to say that they're a bad organization. They do a lot of good stuff, like pay me occasionally, but they are a private company, and as a private company, they need to turn a profit. They're owned by Macmillan, which is one of the biggest publishers in the world. So they need to make enough money that they're ahead of the game. And of course, if you're going to switch over to open access, they're going to do that by charging the researchers the open access fees. I don't have a good solution to this, but if you look at society journals or you look at PLOS, they're often also turning a healthy profit. And why is it the job of the researchers to pay for somebody else to profit from them publishing their research? So there are expenses, but you could probably do this for less if you could figure out how to do it with some sort of self-organizing system. And I gather that's what the AI researchers are doing.
Does PNAS charge?
Mm-hmm. Mm-hmm. And if you want it to be open access, it's more.
It's a national. It's run by the government.
I'm not sure it is.
Proceedings of the National Academy.
Yeah, but
the National Academy is a government body.
It's not a government thing.
They're appointed by.
The National Academy of Sciences is a complex organization.
Yeah, it's complex. It's complex.
Yeah,
I agree with that.
They're a government organization. They're also sort of a private club, and they do both things.
That's how they're selected as a private club. But
club.
they serve at the bidding of Congress. That's the way they were established.
That's
their job is to advise Congress.
I never understood why they have to charge. If it's a government organization, exactly. Why do they have to
That's
charge?
what I was raising.
It doesn't make any sense.
Right. Why is it like NWR?
We don't have any high-profile journals that are free.
Free to publish in?
Mm-hmm.
What do you mean by high profile? So you
Science, Nature, Cell.
No. So virology, you can publish in virology for free. And that's why I often send a paper there, because it doesn't cost anything. And you don't pay anything extra.
And they make their money through advertising?
Well, unfortunately, they're part of Elsevier.
Oh, Elsevier. Mm-hmm. They make a lot of money.
A lot of journals you can publish for free if you mean the author's paying. So
your grant pays for it. Yeah.
So you do your research and you submit it to a journal one of the Elsevier journals, you're not paying them a page charge, but they are charging a subscription fee.
Right.
So it's just a question of who's getting charged.
Which most libraries pay. And
yes. And that comes out of the institutional overhead fee, which comes out of your grant.
Exactly.
So, Alan, I have a question. So, usually the subscription fees are not too bad, like for science, right? Nature.
For science, for nature, those are fairly reasonable. The problem comes when you're an institution and you have a library that needs to subscribe to every Elsevier journal, and they're going to get charged gazillions of dollars to do that.
That's right.
So now, if you go online and you're not going through a university computer, you want to look at an article. They say it's $45. Why does it have to be $40? Why can't it be $6?
Exactly.
Why does it have to be $45?
You'd have to ask somebody way above my pay grade that question, and they will not give you a good answer.
So if you can subscribe to Science for $90 a year, why do you have to pay $30 to see one article?
Because they want to discourage you from reading one article. They want
Correct. That's exactly what they want to do.
But
I wonder if
especially
anybody
when you
ever
could email the corresponding author.
Yeah.
Yes, you can certainly do that.
The thing is that Science is one of those weird peer-reviewed journals that you can buy at the magazine store.
It is. It's on the.
If you can find a magazine store.
In those train stations, they still have.
They're open access in that sense.
And how much would you pay for an issue?
I think it's $5 or something.
No. It's more
than that?
12 or 15.
Oh, it's more.
It's about the same price as the New Yorker.
The last time you
magazine.
bought a New Yorker, no, that's big number circulation, so they can charge less for that. But I would bet Science is anyway. Yeah, you can always email the authors, and they usually will send you the PDF.
That's right.
So if you encounter that, I have never had anyone say you can't have a PDF.
Right.
My article.
Hell, we send a whole book like that.
Was that? Yeah, you give away parasitic diseases, 6th edition.
That's right.
Of course, you have a subsidy for that, right? Not everyone has a subsidy.
Yeah, it's pretty limited, but we do have a subsidy. You're right.
But good for you.
Yeah, good for us.
All right.
If someone wants my PDFs, I'm flattered.
Yes. Well, I published a paper in virology last year, and it's open access and no publication charges.
Right.
So I think that's a good for virology.
Yeah.
But if you're a new assistant professor and you want to get tenured or get grants, you're not going to publish there. You want a higher profile. And this is the problem that we're facing, how to get around this, where the virology paper may be awesome, but whoever's looking at you to hire, they're going to say, virology, I don't want this person, but maybe the paper is awesome.
Maybe some marketer at this journal said AI has risen to the level of now every university has to have a department of AI, and therefore we're going to produce their journal because we can now make money on it. Because in order to get tenure in AI, you've got to publish in our journal.
Tough problem. Just like we've encountered many tough problems today. Polio, eradication, publication. Nipah. Well, there you go. Let's do some picks of the week.
Hey, good idea.
Brianne, what do you have for us?
Right. I recently found out about this site thanks to Twitter. This site is called BioRender. And they have some image templates and icons that you can use for scientific illustrations. So you can set up a free account and basically you'll get something that looks a little bit like a PowerPoint slide. And then there's just this list of icons. So you can put in cells or antibodies. You can have, a healthy mitochondria or a damaged mitochondria. And so I am very, very, very challenged with art. Sometimes my slides from my lectures are a little tough because I'm using, PowerPoint squares and circles to draw things. And these are all correct icons.
Sounds good.
But it's not free, right?
It is there. You can get a free account. Yes. So I've been using it for free. They have paid accounts as well, that some people use. Some people I think are using them to get, icons to use, for figures for papers, like a model figure.
Yeah.
And if you want to get something that's high resolution enough to use for that, then you need to do the paid version.
Yeah.
But for my lectures, they're great.
So the free version for your lecture is good enough, right?
Yes, it is.
That's good.
I was just looking at the pricing page, and they have $35 a month. You can get permission to publish in journals and some other stuff, and then you can put your whole lab on it for $100 a month. But if you're just putting slides together for lectures or giving talks, you have permission for educational use for free. So this is very cool.
Nice.
Yeah.
Here's a cool one with all the viruses.
Yeah.
Huh.
Yeah, I've been really impressed by everything that I've found. It's been very useful.
It's an interesting model. Give a little more for some money.
Yeah.
See how it works. Because that $35 a month is kind of a little bit high, although if you have...
For industry, yeah.
The $35 will get you a high-res download for publishing.
Right.
Well, it's cool. It's an interesting idea.
Yeah.
But I've been impressed, and they help me a lot.
Good. That's pretty neat.
Cool.
Dickson, what do you have?
I have the latest proof that Einstein's theory of gravity using quantum mechanics, which he was not a believer in, was actually correct. So what they did was they focused in on a star which revolves around the supermassive black hole at the center of our galaxy. They've been tracking the path of this star for the last 25 years. Can you imagine that? And what they realized was this star will pass within 20 million kilometers of the supermassive black hole. So there's enough gravitational difference between where it was and where it's going to be to affect the color of the light coming from the star based on redshift of gravity, which is the same as redshift of, further away you are, the redder the star. And they saw it. They actually saw it happen. They watched the star as it made its fast approach and then back out again on its elliptical orbit. And they measured the emission spectra for the star, and it exhibited a gravitational redshift.
Amazing.
Has anyone
fantastic.
Has anyone gotten Einstein's thoughts on this?
Well, they tried. They're still waiting for a response. But I thought this was phenomenal. That's a 25-year experiment. Now, what if someone decided at the moment that they said, hey, you know what? If we could follow this star, we could get a PhD thesis out of this.
I'm thinking of how you get funded for this. The goddamn panels would say, it's too long,
That's
too risky.
This was not something that required, daily input by somebody. I think it's just you check on this periodically,
But you should see how they did it though. They used the very large telescope.
Yeah, it costs money to get on that.
To put 4 big telescopes together to equal 1 telescope. And there's a video on this that you can download. I didn't list it, but it's really amazing. I was just absolutely.
Do you subscribe to SciAm?
No.
You just look online, right?
Yeah, I got this one by just, actually this came up on Yahoo News. I hate to say it. I do that for humor in the morning. And occasionally there's a real article, and you can tell by whether it's a sponsored or not. And this one was not. And I went to it and I found the videos and everything else. So you'll really enjoy it.
Alan, what do you have for us?
I have something that's actually this is something for Senator Cruz and his colleagues to take a look at.
Right.
So this is from the Flying magazine that I get, and this is a fellow who's, you can ignore most of the text, which is about the flying aspect of this, but the intro to it is kind of interesting. He was living in Wyoming several years ago, and he wanted to go around taking aerial photos of the glaciers of the continental U.S. because he was living in the middle of them. And he realized he was going to move to Europe for several years, and he realized that this was going to be his last opportunity. He basically had a period of a few months where he could do this when the glaciers are not covered by winter snow, so you could actually get pictures of them.
Right.
And he knew that by the time he moves back from Europe several years from now, predictions are that most, if not all of these, won't exist anymore. Mm-hmm. The upshot is that these may be some of our last low-altitude aerial images of these glaciers, because they're hard to get to in this way, and they're pretty cool photos that you can go and look at. He has a book out that I haven't bought, but he's obviously promoting that as well, where he's got larger format versions of these. But I just thought it was a really, one of those small examples of climate change just put in perspective that he's thinking that if he's not back in the country soon enough, these things might actually not exist anymore.
Right.
Wow.
Alan, my wife and I were in Alberta. We were in Jasper, and we sat for an hour and a half and observed the glacier called Angel Glacier. And Angel Glacier, in that length of time, lost one of its wings. It actually broke off.
Wow.
As we were looking. Everyone else was astounded. I mean, it was a huge piece of ice, and it just fell right off the damn thing. So, the Columbia Glacier is the best one, I think.
That's what I was going to say.
It's the best one because they have markers as to where the glacier was by year.
Yep.
And now you need a bus to get to the glacier from the road.
We did that about 5 years ago. You had to get in a bus, right, and go up to the glacier. And it used to be right near the road.
Correct.
Right?
Wow.
So
it's up in Canada.
That's right. It is.
In Alberta, right?
It is. It's in between Jasper and Banff.
But once you walk on that ice, it's amazing.
It is. And you drink the water.
You can drink the water.
Thousands of years old. And
amazing.
It truly is. There is one place on Earth where the glaciers are actually growing, and that is a central portion of Argentina, where the glaciers have not been affected by climate change. It's the only place that they said, everything else is shrinking, and this place is staying.
There you go. That just shows.
Right. So if Ted Cruz wants to really talk about that, he can talk about Argentina.
Yes.
Yeah.
Rich, what do you have?
This was sent to me by our friend Nassine because it involves his son-in-law, Mark Settles, who I've in pics previously have highlighted. Mark is a plant scientist at the University of Florida, and he's also a space nut. I had a pic a while ago of this project that NASA does where it tries to simulate a Mars mission.
Right.
And they have teams of people who basically go into isolation, a containment facility that is a mock-up of what would be a Martian ship to study what it would be like for humans to try and live together under those circumstances for an extended period of time. But at any rate, Mark has sent a project up on the latest resupply mission to the space station on growing algae in space for a variety of reasons. Cool. And so I picked that mission. This is the SpaceX CRS 15 mission. CRS stands for Commercial Resupply Mission. And so I have a U.S. National Lab site that describes the mission, and if you pan down on that, you can see Mark's project. And I also, just for quick reference, had a wiki link to the CRS 15 mission. It's really actually quite interesting. The bulk of, the over half of the tonnage that went up was science investigations.
Cool.
Got 2,700 pounds of science investigations and 200 pounds of crew supplies.
That's an Elon Musk company, right?
Yeah.
So the rocket returned to Earth and landed.
I don't know that this one did that. It may very well have, because that's his gig now. Mark has had postings. He's a Facebook friend. Mark went down to see the launch, right? And he can follow the experiments on the space station and advise as it goes along. It's pretty cool. One of the messages here is that if you dig space, even if you can't be an astronaut, you can participate.
That's right.
It's cool.
That's right.
And I also am fascinated by the fact that SpaceX has actually apparently succeeded in commercializing space.
Yeah.
That's right. It's incredible.
Yeah. You're right. And the Virgin Atlantic deal has gone well also, by the way. Mm-hmm. They just had a successful launch of one of their vehicles that's going to the edge of space to give you a view of what it's like to be in space without actually having to go into orbit.
Cool.
Cool.
Yeah.
All right. My pick is a newly released video from ASM Microbe, which was recorded in Atlanta in June. It's True or False with Michael Schmidt. It's a lovely little piece he did in front of the audience where he basically debunked microbiological myths, the 5-second rules, whether urine is sterile, the effect of cranberry juice on urinary tract infections. He's great. And then the audience peppered him at the end, and he's really good.
A lot of urologists should watch this one.
Yeah. So it's a nice little video. produced by ASM and Ray Ortega. And we have a listener pick from Jolene. Greetings, TWiVerinos. My husband forwarded me this very sad article about vaccination behavior in pet owners in the UK. In it, the British Veterinary Association issues a statement that dogs do not develop autism. Apparently, pet owners there and in the U.S. are declining immunizations over just this concern. I was relieved to see no indication that the rabies vaccine is also being given less. Guess I will add pet owners to the list of individuals that it is a good idea to bring up vaccines with in regular conversation. From the household with 3 pets and 2 humans fully vaccinated. Is crazy.
I have to say that my first reaction to this was, how do you tell if a dog is autistic?
Autistic, yes.
Yeah, I just think it's crazy. And
it refuses to fetch.
If you don't immunize your pets for parvovirus, they're gonna die. That's a very bad infection, and so
or distemper.
Come on. The thing that really struck me was this vaccine thing we did in Irvine, Rich, where they have some remote island that got measles vaccine. They never had a case of autism. Remember that, Rich?
Right. Yeah. Right.
Never a single case. So this is crazy. Come on, take care of your pets. This is ridiculous. My home, 2 adults, 3 kids, fully vaccinated.
Right.
Yep.
And
Probably
1 dog. You have a dog too, right?
2 dogs, fully vaccinated. I walk around barefoot, and my wife said, "There's poop over there. Go get it on the lawn." And I said, "I don't want to walk on the lawn barefoot because I might get cutaneous larva migraines." I can't repeat what she said to me, but our dogs are treated regularly because so they're not going to be shedding any. But I thought of it.
That's great. That's absolutely great.
Yeah. Whenever somebody tries to tell me about vaccines, I always point out that I had to get a smallpox vaccine to work with vaccinia in a lab before. And I was actually born a few days after smallpox was declared eradicated. So I have been vaccinated against a virus that has not been on Earth at the same time as me.
Cool.
Right.
So I'm okay with vaccines.
Well, it's somewhere on Earth, right?
Well,
it's still out there.
It's somewhere on Earth. I still have my smallpox scar. Me, Dickson,
I have one.
Rich, Alan.
I have one too.
You have one too, yeah.
Mine's very hard to
faded. Mine's sort of faded.
Faded?
I think it's faded.
Can't get my shirt up high enough, but I'll check tonight.
Right.
All right, that's TWiV 504. It's nice to be back in the usual studio business.
Yeah.
I like being on the road and in front of audiences, but this is good too. You can find it at microbe.tv and at any podcast app that you use on your phone or tablet or any mobile device. You could search for TWiV and you will find it. And please subscribe. That really helps us for you to subscribe to it instead of saying, I'm going to listen this week and download that week. We get better numbers and that helps us with funding the show. If you like us a lot, please consider supporting us financially. You can go to microbe.tv/contribute, where, among other things, you could go to Patreon and give us $1 a month. That would be really great. And send your questions and comments to TWiV at microbe.tv. Dickson Despommier is at parasiteswithoutborders.com. Every week I have to decide which one.
It's all your fault. You're managing them all for me.
The one we're working on, as soon as it's done, we'll bring it up, but not today. Okay.
No,
Parasiteswithoutborders.com. And if you want a little artsy site, thelivingriver.org or DespommierPhotoArt.com.
Yeah.
Haven't mentioned that one in a while.
That's true. That's true.
We built all those together, right?
We did. We had a good time.
Well, I yell at you a lot. So.
I turn a deaf ear.
Thank you, Dickson.
You're welcome.
Brianne Barker is at Drew University. She's BioProf Barker on Twitter. Thank you, Brianne.
Thank you. This was wonderful to be here.
You should use this in your class so that they can hear you on the big media, you know.
That's right.
I have used it before. My students listened once for an assignment, and they said it was creepy to hear my voice in their dorm.
Creepy.
That's weird. I guess a prof in the dorm is, that is pretty creepy.
It's worth 2 in the hand.
So I know a friend who is he was formerly a provost at Brooklyn College. He just moved to Hunter. I went to have lunch with him a couple of weeks ago, and he said, I go home. He's got a Brooklyn accent. I go home and I come in the door and I hear your voice because because his wife is a scientist and she plays the podcast in the house. Says, "Why do I have to come home and hear Vinny's voice?" Oh, well, sorry. Rich Condit is an emeritus professor from the University of Florida at Gainesville. He lives in Austin, Texas, but he's currently in Bend, Oregon. Thank you, Rich.
Almost the last blockbuster.
That's true. That is true.
Always a good time.
Thank you for interrupting your, I don't know, it's not a vacation because you're retired, but it's a thing. You're away from home, and
it's true.
You're there with your family, right?
Yeah, well, they're all, actually, they're all out doing stuff now. I'm just here with my wife, but there's a lot of family in the area. Actually, there are 4 sisters, 3 of her sisters who are here right now. So they're all off having a good time.
All right, now you can join them. Thank you. Alan Dove is at turbidplaque.com. He's also on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
And I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASM for their support and Ronald Jenkins for his music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.